All right, good morning, everybody. It's a pleasure to introduce Dr. Chad Myers. Chad is an associate professor in the Department of Computer Science and Engineering, and he received a triple degree in computer engineering, mathematics, and physics from the Southern Methodist University in Dallas in 2002, and then went on to do his graduate work in computer science at Princeton. And during that time, he started a collaboration with Charlie Boone at Toronto. And Charlie, as some of you might know, is the father of the robotic genetic interaction screens in yeast. So he invented the automated system of doing genetic screens. And Chad provided the whole bioinformatics pipeline for that. And when Chad came here in 2008, he continued this collaboration, but he is also highly collaborative with researchers in the Cancer Center. He is a member of the Genetic Mechanisms program. I'm fortunate to be one of his collaborators, and we share our love for yeast. But so recently he has moved on to transfer all what he knows about yeast to the human system. So he's doing both genetic interaction screens and chemical genetic screens in human cells, and we're part of that. He has received throughout his career many prestigious fellowships and awards. He has collaborated with Carol on breast cancer, and received an NCI Up for the Challenge Stimulating Innovation Award, and the list goes on and on. So thank you for being here and talking about translating insights from yeast to discover genetic interactions in humans.
Thank you, Anya, for that really nice introduction. I'm happy to be here to talk to all of you, and I'm happy to stop, so don't feel like you have to keep your questions till the end. This can be more interactive. I'm happy to explain, especially since I'm coming from the more computational side, so if I go over something that doesn't make sense or you just want to explore in more depth, please feel free to stop me at any point. So I'm going to talk, so we've been working on genetic interactions for basically since I started my lab here and before that, and a lot of that work started in yeast, as Anya nicely described, because that's really the only place 10 years ago that we could introduce combinations of mutations and understand how they affect cells. And what I'm going to talk about today is a story that really evolved from that, taking what we learned in yeast and trying to apply that to understand how mutations in humans predispose individuals to disease. So this is really a project about germline mutations and combinations of germline mutations, germline variants that we think are important in many different complex diseases, including cancer. So I'll sort of focus on breast cancer, since that's one of the applications that we've talked about. So before I begin, just a disclosure, so we have recently started a company, Bridge Genomics, that's negotiated a license for the algorithm I'm going to describe in this talk, so just in full interest of disclosure, and this is part of my conflicts of interest management. So happy to talk more about that if you're interested. I want to also begin by acknowledging two really important players in everything that I'm going to talk about today. So Wen Wang, who's a postdoc in the lab, she was a graduate student here in Minnesota. She really spearheaded the development of a lot of the computational methods. Early on, this was also a major collaboration with Gang Fang, who's now at Mount Sinai. He was a student in Vipin Kumar's lab at Minnesota. So I'll come back to these people at the end, but really the algorithm I'm going to talk about is a result of years of work by both of these students and now faculty. Okay, so the backdrop for my talk is this plot. Some of you have probably seen this before, but there have been thousands of genome-wide association studies. So again, I'm talking about germline risk here, and one of the ways that we identify germline variants that predispose individuals to disease is genome-wide association studies. And the NIH has captured these associations quite well. There's a database that tracks all of the associations that have been made across many different human traits. And what I showed you is a video that captures from starting in about 2006 up to 2018, the progression of our knowledge of different variants in the human, different loci in the human genome that are associated with many different traits. So this includes mostly disease traits. Essentially, these are individual SNPs, single nucleotide polymorphisms for the most part, and we know that they're associated with these diseases. And you can tell we went from relatively few of these in 2006 to basically it fills up this slide today. So in that sense, this approach of, and I think most of you are familiar, but maybe just to make sure we're on the same page, the way genome-wide association studies work is you collect cohorts of individuals that have a specific disease. You typically try to make this homogeneous in terms of ethnic background, exposures, many other factors, control as many as you can, collect a control group that otherwise is similar except for the occurrence of disease, and then try to identify loci that are enriched in the disease group or depleted in the disease group. So that's, when I say GWAS, that's basically how it's done. This started out with people in the cohorts in the size of hundreds or thousands. Today, people are pushing these cohorts to be tens of thousands, hundreds of thousands, as is possible. So I think we have people in the room, for instance, that work on pediatric cancers. There are many cancers where those kind of cohorts just aren't even feasible to collect. We don't have enough individuals that get these diseases. And that's obviously a major challenge for these kind of statistical methods. Okay, so from one perspective, these approaches have been highly successful. Look at this slide. One of the sort of downfalls of this whole approach has been this problem that people refer to as missing heritability. There's a number of high-profile papers that discuss this, and really there hasn't been a conclusion about why is this the case. So let me describe what missing heritability is. So the concept is this. We have a number of loci for most diseases that have been studied. You can find a handful of loci up to maybe 10, sometimes even hundreds of loci in the genome that are associated. The issue is if you then take those loci that we know about and, say, build a model that predicts who's going to get the disease, for the most part, those models fail at predicting pretty badly. So they're non-random. And we can actually measure that. So statisticians can measure, statistical geneticists can measure the proportion of the heritable risk that's explained by a certain number of loci in the genome, or a fixed set of loci in the genome. And what this means is that we know these diseases are genetic. We can figure out how much of that disease risk is explained by genetics. And these numbers show you, these are actually fairly old, but the story is very similar today. All these diseases had this many loci discovered at the time. This is the fraction of the heritable disease risk that could be explained by the loci that we knew about. And notice these numbers. In the case of macular degeneration, the story is quite positive. We can explain something like 50%. That's probably more like 70 or 80% these days. Most of these diseases, so type 2 diabetes, high cholesterol, there's clearly a genetic component to them, but we can explain maybe 10%, 20%, well less than half for most diseases. And that still remains true today. We've expanded the number of loci for some of these. I think height is an interesting exception. I think the latest number has gone up to hundreds of loci that affect height, and together those explain something like 60 or 70% of the variation. So height we've gotten better at explaining. Most diseases, especially complex diseases that affect large fractions of our population, there's still a big gap. We know they're genetic. We try to find some factors. We do find some factors, but when you put them all together, they still don't explain most of what we see. And there's a lot of reasons why this is probably true, and this has been debated a lot in the literature. One is that all of these studies, most of you would probably know, GWAS, for the most part, studies common variants. So it's 5% frequency or higher in the population. It could be that there's many rare variants that aren't even captured on these chips that that's why people get disease. And if we just looked at rare variation, we'd actually see it. And there have been success stories there. So in autism, this may be the biggest example of that. There have been really rare but high impact variants that have been associated with autism. I would say still, even in autism, that hasn't explained everything that's been seen. So that's clearly one of the factors at play here. GWAS is not powered to look at rare variants. I think another interesting one that's been debated quite a bit is the concept that many of these diseases are so polygenic, it's not about a small number of loci that contribute. It's about risk that's spread across 100 loci in the genome, and they're all such small effects individually that you can't really build a model that's based on just a few. And this is I think it's a more depressing explanation. It says that we actually can't pin this down very well to genetic factors. The explanation I'm going to explore today, we've been thinking about this for really a decade now, is the concept that it's not just about which individual variants you inherit, but it's about combinations of variants that you inherit. And all of the paradigm that's been applied to study this problem, for good statistical reasons, basically has ignored that contribution. So they basically, the whole research methodology around GWAS studies is designed to ignore genetic interactions. And we think that that may be at least one of the explanations for why we're not able to explain disease. Okay, so let me explain why this is a difficult problem, why really the field has tried to sidestep it, because it's a major issue. So, the current paradigm is basically to take an individual variant, let's say this A to T change or T to A change at this single locus in the genome, collect a cohort and test for association. The association tests are simple. Basically, you say who has this variant, who doesn't have the variant, and then you look at ratio of disease in those populations. So it's a really simple statistical test. As most of you know, these are called genome-wide for a reason. You do that test not just once, but you do it for 500,000, maybe 1 million loci. If you filtered it for what you know in advance, maybe a couple hundred or 1,000 loci. So this is a multiple testing problem. So when you do this many statistical tests, what you have to do is correct for the significance of those, because most of you, I think, probably know the multiple testing issue. So if I do 100 tests, I set a 5% significance threshold. 5% of those tests are going to be false just because I've done that number of tests. So this becomes a huge issue if you're doing 500,000 or 1 million tests or even more, because you have to dial up your significance threshold to such an extent that you don't have the power unless your cohort sizes are big to see that. So that's even a problem for single variants. Now, let's think about what happens if I try to test combinations. Let's say I'm not going to just test what you have at this location if it's an A, but I'm going to look at a different variant location X, and I'm going to look for not just this combination, but I want to look for all possible combinations. I don't have any knowledge of this going in, which is really the state we're in. Now what I've done is taken a problem that's already hard, let's say a 500K tests. I'm going to do 500K choose 2 tests, so we're getting into the 500 billion tests range. And 500 billion, if you do any, play whatever statistical games you want, we're going to be underpowered to detect anything. You need cohort size. It's estimated a cohort size of maybe 500,000 and a pretty large effect to have power to detect that with a standard pipeline. So using a standard GWAS approach, it's actually quite hopeless to try to detect interactions without some knowledge going into where those interactions might be. So any questions so far? Everyone's with me? So this is the sort of statistical challenge that we're trying to address. Okay, so I'm going to take a detour through yeast, and as Anya said, yeast is actually something we still care about a lot. It's an interest we share together, and yeast is still teaching us a lot of important things. So I'm going to give you a sense. So basically, so this is Saccharomyces cerevisiae. This is a thing that is the organism that's used to brew beer, bake bread. And as Anya mentioned, what's important for this talk is that almost 20 years ago now, actually more than 20 years ago, Charlie Boone, Brenda Andrews, they developed technology that they could actually go in and efficiently knock out combinations of genes. So if you think about what's going on in the human genome, I have about 1 million variants relative to any of you. And for us to tease out what is the effect of a combination of 2, that's a pretty messy setting. But if I can take a genome and introduce 2 and only 2 changes and then analyze the phenotype, we should get a much better sense for the rules that govern these kinds of interactions. And that's exactly what we can do in yeast, and not just for a single combination, but we can do this for hundreds of thousands, millions of combinations of mutations. And that's basically what we've done for the last 10 years in collaboration with Charlie and Brenda at University of Toronto. And so this is enabled by robotic approach. This is, I think many of you probably know about synthetic genetic array approach. And really this experimental approach motivated techniques like it all across the tree of life. So we're now, as Anya said, diving into applying CRISPR screen technology to do the same type of thing in human cells. A lot of how we approach that is guided. The genetics are completely different the way you do it, but a lot of how we approach that and approach those screens and process data is motivated by everything we've done in yeast over the last decade or so. Okay, so I'm not going to talk a lot of detail about the basis of this, but the phenotype that we look at is a plate, an agar plate. Each one of these different each one of these different dots is a colony of yeast growing up over 2 days or so. And you can tell that some of these, so each group of 4 shares a different double mutation, so a precise set of 2 genes. And most of these are deletion mutants. Some of them are conditional mutants that we can actually start to look at essential genes as well. So combinations of mutations in essential genes, not just full deletions. Okay, so that's what the sort of raw experiment looks like. In my lab, we've worked a lot on translating this into quantitative data. If you're interested in how that works, you can read this paper back in 2010. But the basic concept is we measure the fitness of each of these single deletions by themselves, fitness being a number from 0 to 1, basically, that reflects the fraction of growth relative to wild type. Essentially, it's a growth rate relative to wild type. And we have a model that most genes should follow, most combinations should follow if they're independent. So statistical geneticists will support this. Basically, if you introduce 2 different deleterious mutations, you expect they should compound each other multiplicatively. And so we're looking for cases that differ from that. So we observe a double mutant fitness. We measure that. We use essentially colony size as a proxy for fitness. And we subtract off what we expect. So we expect 2 sick mutations to compound each other. But if we see a case, for instance, where 2 partially deleterious mutations actually in combination kill the cell, this is greater than we expected. Or vice versa, we could find 2 mutations that maybe cancel each other out. That is, one is very sick, but in the context of another mutation, you see genetic suppression. So you can see both directions here. You can see interesting cases where it has a more deleterious phenotype than expected or a less deleterious phenotype than expected. And both of these are informative. Be happy to talk in more detail about those and what we can learn from them. So that's the basic idea. We've made a lot of progress over the last decade or so. This technology really was mature maybe 10 years ago. Charlie and Brenda's labs turned the crank on this, and we produced about 23 million double mutants in a single strain of yeast. About 1 million of those cases, and this is all described, the latest iteration of this is described in this paper in 2016. And at this point, in terms of a standard lab condition, a single standard lab condition, we're effectively done. There's no more reagents that we can use to combine other genes. There's still interesting things going on in that front. The main focus on the yeast front now is what happens if you do this across different conditions. This was all done in a single lab condition. So we have a project that's ongoing right now, which is let's do miniature versions of this map, but in 15 different stress conditions. And let's understand how combinations of mutations look across these different environments. So that's an ongoing direction. Of course, there's analogies there to what happens if I look in different human cell types, for instance. So this is another project that actually is a collaboration with Anya that's funded by NSF, is we're looking in we're doing the same type of screens in human cells using CRISPR, but in different cell type backgrounds to say how are these networks different. And the idea is that we can guide how we approach this problem in human cells. There's a lot more complexity. Which cell type do you pick? What you learn in this cell type, does it inform what you do other places? So that's a major interest that's going on right now in yeast and in human cells. So back to the story here. So we've constructed about 23 million double mutants. About 1 million of those exhibit cases where there actually are interactions. There's differences between the fitness you measure in double mutants and what you expect based on the combination of the single mutation. So, and I'm not going to talk a lot more detail. I'm going to skip that slide. I'm not going to talk a lot more detail about what we've learned from that. I'm going to tell you really 2 key things we've learned as it motivates what we've done in the human genetics problem. One is basically that these are relatively rare instances. So genetic interactions happen about 5% of the time. So combinations of, these are mostly whole deletions, combine to cause a greater than expected or less than expected phenotype about 5% of the time. So it's quite rare. I would contrast that with something like physical interactions. So there's been a lot of work to map protein-protein interactions, for instance, using mass spec and other types of approaches. Those networks are much, much sparser than this even. So in yeast, there's something maybe like 50,000 physical interactions that are characterized. So the number we're talking about is about an order of magnitude more. So when you think about which proteins are physically interacting with each other, the ones that show genetic relationships like we're talking about here those relationships are much more frequent and more complex. I would say harder to interpret, partially because of that. This figure is showing you basically across all these pairs that we've screened, most of them actually fit the model we expect, so they fall right at 0. That's this peak here. There are interesting cases that exhibit, this is the difference of what I observed and what I expected in the combination of the double mutants. Interesting cases out here, and this is just a log scale on the y-axis. So you can see this tail out here are cases that are sicker than expected. This tail up here are cases that are healthier than expected. And just to show you, so we can detect about a 20% difference in fitness is about where our resolution is. Okay, so I will say a couple of key lessons we've learned that motivate what we're doing in humans. So one interesting view of this data is this is just a map of different functions in the cell, so different sort of biological processes or neighborhoods that yeast cells accomplish to carry out life. And it's organized symmetrically. So there's a bunch of genes. I grouped all the genes involved in metabolism here. You can see the same order appears here. And what I'm showing you here is, or Elizabeth and Ben made this plot. Basically, I'm showing you the density of interactions. That is, what's the frequency of pairs that interact when you combine genes from this neighborhood on this axis and this neighborhood on this axis. And just looking at this visually, you can tell there's a major trend, which is that the diagonal tends to be much bigger than the off-diagonal. And the reason for that is that if you combine two genes that are in broadly related areas of biology, and these are pretty broad, look at, I'm talking metabolism or transcription, chromatin organization, DNA replication repair. These are not individual pathways. They're more like larger processes where there are many different pathways that contribute to that same function. That's where you see the highest density of negative interactions. These are synthetic 6, synthetic lethal type interactions. So already you can tell this is sort of a guide that tells us where are these interactions most likely to occur. There are interesting cases that are off-diagonal. So I should say there are interesting processes like chromatin modifiers. You can see there's a set of these that interact pretty broadly. So you combine a mutation in an HDAC with many other genes, you'll see a strong phenotype, even when there wasn't strong phenotypes to begin with. So there are major exceptions to that, and some of those are quite interesting. Those form the hubs of these genetic interaction networks. But we're going to leverage the principles that emerge in the sense that the related genes tend to interact with each other more than unrelated. Okay, so I will just make one other point about the yeast network before we move on, just to say that one really powerful thing that we've done and others have done in the yeast field, and this actually is really what's motivating a lot of our work in human cells, is that you can treat these interaction profiles, that is a gene's set of interactions with the genome, as a fingerprint for what its function is. So what I mean by this is a cartoon data here, but and it sort of looks like a gene expression profile if you think about it. But what I'm talking about in each square is whether or not when combined a mutation in this gene and gene A and gene E, for instance, had a genetic interaction or not. So it's not about expression. It's about was there a phenotype when you combine these mutations. And this profile for a given gene, then I can focus on a gene A's profile or a gene B's profile. And it turns out that it's quite difficult to interpret. A given gene might have 50 or 100 interactions across the genome. Understanding precisely why mechanistically that gene interacts with all those other genes is actually there are very few cases where people have gone in and understood why that happens. It often makes sense, but mechanistically it's hard to pin down exactly why that happens. Yes, go ahead, Brian. Yes, perfect.
We need a microphone so they can hear you across the way.
There are some genes that regulate other genes.
Yeah.
So that when you look for an interaction, it may not be gene A is interacting with B. It's genes that A regulates interacts with B. How do you account for that distinction?
Yeah, so this is where this is the difference between direct physical interactions and functional relationships. So in the case that gene A regulates a set of downstream genes, B, C, D, what we expect is that all of those exhibit the same interactions with anyone else in the genome. If A is its role is basically to regulate these. If you miss A, it's the same as the phenotype is missing any of these downstream regulators. That's assuming they're all essential for whatever process is causing the phenotype. So those, in some ways, the physical interactions, the physical connections, these regulatory connections, are orthogonal to what we're mapping. They define these modules, and what we map are interactions among those modules with other modules in the genome. So for instance, A, B, and C would be in a co-regulatory module. It might include the transcription factor plus the downstream targets that are regulated, and together those accomplish some function. If the regulator's not present, those genes don't get turned on. If those genes aren't present, they don't actually accomplish what the goal of that pathway is. But what we see is that they share exactly the same interaction profile. So the phenotype you see when I combine any of these mutations with other mutations is very similar. And that's what we extract from these data a lot of times. It's this modules show very similar relationships. Does that make sense, Brian? Okay. So yeah, and this is where it's very complementary. So a lot of work has been done by us and others to combine physical interaction type data. It might be transcription factor, target relationships, or direct physical interactions between proteins. Combine that data with the kind of data we get here because they're very complementary. Okay, so basically you can take these networks then and cluster them. So we basically just take profiles, find pairs that are very similar. We turn that into a relationship network. That's what I'm going to show you in a second here, where every gene's connected if they share a highly similar profile. And that's what this map is here. These are basically all the genes in the yeast genome. And we use a spring model layout algorithm. All it does basically is draw things. Essentially, every edge that has a relationship is modeled as a spring. You shake this up. This is how that relaxes. And you get these tight clusters that reflect a bunch of genes that are all very tightly connected with each other because they share a lot of similarity in who their interaction partners are. And you get others that are loosely connected. So what I just described is basically the same data game that Facebook, any of these companies that are using big data, they collect data sets, they use them as profiles, and they take what they know about some individuals and translate that to others that are related in the data. And that's exactly what we're doing here. I can label these neighborhoods based on some of the genes I know their functions. In yeast, it's about 3/4 to 85% probably are pretty well characterized. And so I can go into these dense neighborhoods and say, wow, this is highly enriched for mitochondrial genes involved in mitochondrial function. And that allows me to extend those annotations to any genes that also cluster in that region. That's exactly sort of guilt by association. This is what we call it in the computational biology community. But that's exactly how people are using data to sell people things and market things and everything else. So you can play the same game with these high-dimensional genetic interaction networks. And this is one reason why we're very interested in mapping these in human cells, because we can go from no understanding to a pretty unbiased global view of how everything's related in a cell. And this is, I should remind you, from a crude phenotype, which is how fast yeast grows over 2 days. So that phenotype is very crude, but still gives us pretty detailed insights about very specific functions that are happening in the cell. Okay, so that's just a sort of broader motivation about why we collect these interactions and why they're functionally useful. So, yep, go ahead. Yep. Yeah, so
you're going to tell us, but what's the super simple phenotype that's equivalent to, size and girth of a yeast colony over 2 days?
Yeah, so I think this is obviously a complex question, and it gets more complex in higher eukaryotes. We are starting with essentially a fitness readout in human cells, and it's not a colony, as you probably know. It's done in a screening context, a population of cells, a competition basically, and primarily looking at cells that drop out when 2 gene mutations are combined, 2 loss of function mutations are combined. So, there are some phenotypes that this is less relevant for that are really important for. So first, it's in a cell line, cell-cell communication, multicellular, which is obviously important for multicellular organisms. We're going to miss out on a lot of that interesting stuff. That said, half the human genome is not annotated at all. We know what the sequence is. We have no idea what those functions do. If I can build a map that's even captures all the processes that impinge on cell growth, which a lot of what cells do sort of that's the downstream impact of that, I think we can contribute a lot to what we know about human cells. So, and that said, it's also interesting, and people are doing this. to use specific reporters. So we're not doing this yet, but you can imagine you really care about this downstream pathway, set up the same screen to sort based on some kind of reporter that's activated. It's not a fitness phenotype at all. It's then, are you able to activate this downstream expression? Does that make sense? And I think all the same approaches could be applied, and you're going to get really rich data about that, the phenotype related to that pathway. Yeah, good question. Okay, so I want to now connect this back to the human genetics problem in the last few minutes. So basically, one of the important lessons we've learned, this is related to Brian's questions as well. So these interaction networks are highly structured, and this is an example of the structure that you see in yeast. So what we often see is that you get a whole, these are complex. This is a chromatin modifying complex, the square-SNF complex, another RPD3. This is a subunit of the proteasome here interacting with DNA replication factor C, a few other complexes. What you see is that if you see an interaction between one pair of subunits that crosses two complexes or pathways, this extends more broadly beyond protein complexes, you typically see all of them or a large fraction of them interacting. So it's very rare to find just a single, and these red ones are synthetic lethal type interactions, synthetic sick interactions. The green on this picture are the opposite. These are positive interactions. And both of these types are highly structured. So the concept, and we can characterize that structure. I'm not going to go into a lot of detail how we've done that, but we've developed algorithms to exhaustively mine for these structures in yeast. And what you find is about 75%, if a geneticist finds a synthetic lethal interaction, just pick one random one out of the network, 75% of the time we estimate it's going to be embedded in one of these larger structures. So this case where that interaction is very specific to those two genes is actually more the exception than the rule. This case where that interaction is instead embedded in a much larger structure, that is, I see an interaction between gene A and gene 1. Typically that's indicative of a larger relationship that if I were to knock out other genes in this pathway, this A gene's pathway, and other genes in combination, other genes in the gene 1's pathway, that's two separate pathways, but if I form combinations that cross that, I'll see exactly the same synthetic lethal phenotype. And this is really the rule that we see in the data, and this is what motivated us several years ago to try to apply what we did in yeast in human populations. And so the concept is really simple. Let's change our statistical tests in human populations from this, which is exactly how all of the GWAS community has really approached the problem, and really from a single variant perspective. You're looking at a single mutation, not even a single gene, but a single nucleotide polymorphism. When you translate that to interactions, we talked about this problem. It's a statistically challenging issue to take SNP A and SNP 1, say. There are a huge number of these combinations, and we don't have the power to do it. What we said is let's instead translate that to this problem. If they're real interactions, we think they're probably structured like they are in yeast cells. I don't think that's a major stretch because all of these biological systems are modular. The human genome is organized into functional modules, into pathways. If there are interactions, if there really are interactions behind these phenotypes, we should see many of these structures emerge. And so what we're going to do is translate the test into these kind of tests, from these kind of tests, translate it into these pairwise tests, but at the pathway level. So we're looking for these kind of structures, and we're letting go of trying to identify individual SNP combinations, at least at the beginning, such that we can have the power to see these effects. So that's the basic idea. So if you take nothing else from the talk, that's the main thing to take, is that we're taking human genotype data. So now I'm all human for the rest of the talk. This is usually on the size of about 1,000 individuals. We've done as few as several hundred, say 500. That works okay. Obviously, our power is tied to how many individuals there are. One important input are pathways. So we need sets of genes as input. That forms pathway A and pathway for gene 1. These are input to this algorithm. So we take gene sets. And what we look for are these kinds of relationships where there's a pathway here, a separate pathway here, and there's a number of weak associations that cross those two pathways. That is, I see some individuals that have a SNP in gene B and a SNP in gene X, I see a different set of individuals that have a SNP in gene A and a SNP in gene Y. But together, if I reflect the fact that, or if I respect the fact that these are coming from two sets of genes that are related, two different sets of genes that are related, I now have the power to fish out this larger association, and I can bring together individuals that have completely different genotypes, but have the disease for the same underlying reason, which is that they have combinations of SNPs in the same two pathways. So does that make sense of the basic intuition behind this? So the tests that we're doing are based on pathways that I've given as input. These are literature-curated pathways. Of course, as pathway curations get better, our approach should get better, so that's a limitation of the approach. Given those pathways, our output is to identify combinations where it appears that there are more individuals than expected that have combinations in these two pathways at once. And that's the kind of discovery we're going to make. Okay, and this approach we call bridge. Okay, so this is basically what I just described, but just to lay it out in more word form. I have to map SNPs to genes, obviously, and then from genes I get to the pathway. So I can essentially associate a single nucleotide polymorphism, this applies to copy number changes as well, with genes in the neighborhood and then pathways that those genes are associated with. Right now we're drawing pathways from a variety of different sources, so KEGG, BioCarta, Reactome. Many of you probably use these on a small-scale basis every day. What we do then is actually we do this test of all pairwise SNP associations. So you can compute this, so it's not a computational issue. We don't have the statistical power to identify any individual SNP pair, but we can, if we step back, we can identify larger sets of them that cross pathways of interest, like I just showed you. And then what we do is test all possible pairs of pathways. So we can't test all possible pairs of SNPs statistically, but using the approach I described, we can test all pairs. If I give them 500 to 700 pathways or 1,000 pathways, I can exhaustively test all pairs of those for interaction. So in that sense, as long as you're okay with being constrained to pathways that are curated in the literature, we can find interactions for any disease that has a cohort that's of a reasonable size. Okay, so that's the basic idea. I think I'm going to skip this concept. If you're a statistical geneticist, I should mention that we have to assume some disease model. So either we have to assume that combinations of variants are additive by expectation, or we have to assume that a combination is recessive or dominant. And we have each of these modes. Whenever we run an analysis of a given cohort, we also think about, do we want to run, in which combination model do we actually want to assume? Do we want to assume that things combine additively? Do we want to assume a recessive or dominant model for variants? Yeah, go ahead, Scott.
Assigning genes to pathways.
Yeah.
When you're assigning genes to pathways, how many genes in the human genome are kind of left out of that curated list?
This is a major limitation. Half the human genes don't show up in these curated pathways, basically. And I should also say, many of these pathways are not nearly as well defined as you would think. They're really gene sets that are loosely grouped together. And this is a sort of a fundamental limitation of the approach. And as we get better pathways, it should improve. One other thing we're working on is, for example, as genomic data sets get better at capturing relationship in the human genome, we don't need to wait for those to be curated into pathways. We can feed clusters, say, from our genetic interaction network in human cells directly into this approach. All it takes is gene sets. I don't need to label what those are, but if I tell you at the end of the day, here's a set of 5 genes that were tightly clustered from our HAP1 genetic interaction screens interacting with a set of 3 that were also tightly clustered there. And now I can discover a genetic interaction in a cancer cohort. It's worth characterizing why are those clustering. So it's a major limitation, and right, I think it should only improve as we improve these curations. Yeah. Okay, so I'm going to talk about some results of this idea in the context of breast cancer. This is a collaboration with Carol Lang. Here, we really teamed up as part of this NCI challenge. There was as Anya mentioned, there was a competition a few years ago that the concept was they focused a whole group on about 5 different cohorts focused on breast cancer. And it was an interesting competition in the sense they said, here's the data. We'll give you access to these data through dbGaP, and we don't have any specific thing in mind, but we want you to do something different. We want you to do something different, and we'll give, there was cash prizes associated with this, no strings attached. And so that, you can imagine the judging of these competitions is fairly unique. How do you judge who did something better when there's not even it's not a prediction problem that's pretty easy to tell who did well. So anyway, so that's a context. That really was what got us started in the breast cancer space. And these are the cohorts that were part of that competition. You can also just access these through dbGaP if you're interested in breast cancer. So it's a mix of a couple European cohorts and Chinese and Japanese, and this single cohort has a mix of ethnicities. So that's the results that I'm going to talk a little bit about today. So let me just start with a single example of something that we find. So first, we can find interactions in breast cancer. We can find interactions in many diseases based on this approach, and I'll try to give you a sense for the robustness and the interestingness of those and some limitations of those findings. So this is from one of the European breast cancer cohorts. Again, we're talking germline mutations, common variants, combinations of common variants. And these are all from SNP chips at this point. And this is a single discovery by our approach. So one of the strongest interactions we found for this particular breast cancer cohort was an interaction, I just noticed there was a spelling error in the steroid hormone biosynthesis pathway. This is from Reactome, and again, that's sort of a high-level pathway. It includes a bunch of different aspects of steroid hormone biosynthesis, so some of you might not be happy with the groupings there. I think it's about 15 or 20 genes, and this is even worse of a pathway. It's acute myeloid leukemia gene set derived from KEG. So this is a set of genes that have all been implicated in AML in some way, and some of the surrounding genes in the neighborhood. So you'll see some of these players in a second. A few common risk factors. Most of these were identified, I think, in somatic mutations from AML, and then some of the genes in those neighborhoods. And so the way we discovered this is basically each one of these lines reflect a connection between a SNP that's associated with a gene in this gene set and a SNP that's associated with a gene in that gene set. And so you can already tell there's a huge number of combinations just restricting to genes in these two pathways. And I'm only drawing edges here where there's a weak association. That is, if I collect all the individuals that have, let's say, a minor allele in both copies of this particular variant, at the same time they also have two minor alleles in this particular locus in the genome, what I'm telling you is that set of individuals has a mild association, that is, they're slightly enriched in this particular cohort. And I say slightly enriched because it's not strong enough for me to get excited about this specific edge for the same statistical regions that I told you before. So I can't tell you, yes, you should worry about these two SNPs. What I can tell you is that there was an accumulation of edges like that were all weakly significant. That is, no single one could pass the statistical tests. But together, there's way too many of them crossing these two pathways for it to be by chance. So, and the numbers are something like, I expect to see 230 at the cutoff that we set. This just gives you a sense for the enrichment. And we see 367. So there's a enrichment, like 1.5x enrichment for how many associations are crossing this. And statistically, these are numbers that now we can get some confidence in. So I can actually have we're powered enough to say, okay, even though I did hundreds of thousands of tests here because I tested all possible pathways, this is significant enough for us to actually say this passes some false discovery rate or even a Bonferroni correction. So this particular case is associated with increased risk. So individuals that have combinations of variants here and here are at elevated risk of breast cancer from this particular cohort. So do people understand what this particular discovery reflects? Go ahead, Logan. Yeah.
Actually, can you clarify what you mean by SNPs in a gene? Are you mean coding SNPs, silent?
Yeah.
Splice certain distance, or what about EQTLs that are distant, etc.? Yeah.
So this is another really important, complicated detail. What we're doing is basically mapping SNPs that are in LD to any gene that's in this case a 50 kb region. So a SNP does not have to be in a coding region. We assume that could be tagging something nearby, or it could be a regulatory SNP. we do not, I think it's one of the next things we'd like to develop is something that captures broader regulatory relationships. So enhancers that might be way more than 50 kb away, but we know what the targets of those enhancers are. We should be able to capture those associations as well. We're not doing that yet. And I think that's, as most of you probably know, in the GWAS community, a large fraction of SNPs are not near coding regions, and so it means you should have some strategy to link those to genes in some way. And that's phase 2 or phase 3 of this project. But it's an important issue for sure. Any other questions about this before I... Okay, so I should mention, I didn't talk about what these bars actually, so each bar is a SNP. The height of these bars reflects, if I do a single association test with that individual SNP and breast cancer in this cohort, and this is a 0.05 p-value cutoff. So this line, so going this direction and this direction is a 0.0, so it's the weakest threshold you can imagine. Most genome-wide significant associations are 10 to the minus 8. None of these individually would be discovered if I just did single association tests. So we're finding pathways that no single SNP in those pathways would even come up on your radar if you're just doing a standard single variant association test. So it says you can only get at these through this sort of pathway interaction. And yet, of course, in breast cancer, it's not surprising you get steroid hormone biosynthesis, potentially a modulator of who's susceptible to breast cancer. Let me just mention that this is the statistic that convinces us that this pathway is interesting. So we can shuffle SNP associations hundreds of thousands of times, and here's the distribution of densities you get across those two pathways. You can see it varies, but it never varies out this far. So this is the statistic that actually gives us a lot of confidence that the observed density of weak SNP associations crossing this pathway is actually interesting. And this plot on the right side tells us what happens if I do SNP pairwise tests. And the most significant interaction I can find here, I could discover at an FDR of 0.94. So it means it's not a discovery. I can't do individual SNP tests because I don't have the power to do that. And we knew that in the beginning. We knew we would not be powered to just do individual pairwise tests. So this is all just to make the point that you can really only discover these associations if we look at the combination of pathway level. Okay, so what I've been talking about is a single pathway interaction. That's this edge right here. It turns out in this cohort, if I'm willing to discover it at an FDR of 0.25, so 25% of what I'm showing you on this slide will not be true. The other 75% has strong support. This is the edge I was just talking about, the AML gene set with this steroid hormone biosynthesis gene set. And you can see I get a number of other pathway interactions showing up. And actually, the thing that emerges here pretty quickly is that this steroid hormone biosynthesis pathway has a number of modifiers that show up. It modifies a bunch of other pathways, it appears. And all of these that are shown on this slide are associated with increased risk. So there are a bunch of other pathways that combinations of variants where one is in the steroid hormone biosynthesis pathway and one is in, say, mRNA processing gene set, one is in a gene set that's associated with type 2 diabetes, those individuals are at increased risk of breast cancer. So that's what this slide represents, is not just one of these pathway interactions, but we have about 10 or so that we can clearly see in this cohort have some support. And one of the, as most of you probably know, in the GWAS world, one of the important questions is, can I take what I discovered in this cohort and replicate that in other cohorts? That is there support in independent populations for the same genetic associations? And often if there's not, that's where it ends. That's sort of the gold standard as to whether what you've discovered is worthy of discussion by the field and worthy of things like functional validation and more longer-term functional studies. And so in this case, this specific interaction, the AML, one of the reasons I'm talking about is because that replicates well across in a second independent European cohort. I should say that between different populations, these interactions are quite different. I'll talk a little bit more about that in a second. But this particular interaction I've been talking about is significant in two different cohorts, suggesting that there's interesting support for that. And I'm summarizing this at a gene level, so I haven't talked about genes yet. Here's a list of genes in the steroid hormone biosynthesis pathway. that we have SNPs in this particular cohort, and the genes on this side are the AML gene set. And you can see I've ordered them by, Wen has ordered them by basically how frequently they're associated with interactions between at the SNP level. So I can see RUNX1 is the top gene in the AML gene set, and you have a bunch of these the steroid biosynthesis enzymes that are forming the top enzymes in this other set. Okay, so there's support in an independent cohort. Some of the same genes actually appear to be supporting that. I'd be happy to talk more offline about these. I should say that some of the strongest interactions are listed here. These are interactions between genes in those two sets that we can see weak evidence for in multiple cohorts. So now we can get to the point where even though we couldn't discover SNPs or genes individually in advance or from the whole set, I can start to prioritize things that have first are associated with these two pathways that were strongly significant and have independent support in two different cohorts. So for instance, rare A is a gene that has been associated with somatic mutations in leukemia, and this is a steroid biosynthesis, I think, corticosteroid biosynthesis enzyme here. And so those, we can actually now start to look at variants that in combination are associated with risk of breast cancer. And this is a long process, not just with breast cancer, but we're applying this approach to a whole set of diseases now. And really the next phase is to get funding to explore these interactions in models of these diseases. And that's a years-long endeavor that requires a lot of focus, collaborators in each of these diseases long-term. And that's something that really, I think given that people essentially assume you can't discover interactions, we need to do that to show that these are functionally, these are actually the mechanistic basis of genetic risk. Okay, so let me just, I'm sort of running out of time here, so I'm going to, let me just say that in breast cancer, there's two other major players that I want to briefly mention. We can also we can run this algorithm in a mode where we combine multiple cohorts in advance and find things that have support up front. And one of the major players that emerges from that analysis that wasn't on the previous analysis of that single cohort is this glutathione conjugation gene set. And this comes down to a set of genes, this GST, so it's glutathione S transferases that appear to be very interesting. So I don't have time to go into detail on this, but if you have any interest in that particular pathway, there's really strong support in many cohorts that these GSTs have a strong association in combination to predisposed individuals for breast cancer. And there is some support, so I should say GSTM1 is one of these glutathione conjugation genes. There's mixed result in univariate GWAS analysis that implicates GSTM1. We think there's a whole bunch of these enzymes that actually support these modifying relationships. So it doesn't come, this is actually driven by many of these GSTs and is supported by at least 3 different cohorts in this set. So if you're interested in GSTs, I would be happy to talk more about that. I just want to wrap up this breast cancer piece. There's also a strong association with GATA3 and a number of the factors that interact with GATA3, which is there's a long history of studying GATA3 in the context of breast cancer. And specifically, so this is what we call a within pathway interaction, where combinations of SNPs in the same gene set, that is a SNP in NFAT-C1, for instance, and a SNP in GATA3 together are associated. There's a whole bunch of these different combinations within. This is the whole GATA3 gene set, basically. And you can see there's a number of these combinations that appear to predispose individuals for risk of breast cancer. So I'd be happy to talk more offline about that if you're interested. So with that, let me want to just sort of wrap things up. We had a lot of discussion in the middle here. So let me just say we've studied 9 different diseases now, and we've discovered interactions in 8 of them. So I've told you a little bit about breast cancer results, but most of these diseases we find pathways that are clearly relevant for the disease and something we're excited to pursue. So let me just end here. I'm going to skip these videos. So we're working on trying to translate these associations into knowledge of new targets. We're working on, I think I just messed up this. We're working on higher order interactions in yeast. We're working on conditions in yeast. As Anya mentioned, we're working on chemical genetic approaches, and we have a big project on using CRISPR to do these genetic interaction screens in human cells, chemical genetic and genetic interaction screens. And I'm going to stop there. So let me just acknowledge Wen and Gang again. This is really a lot of all the methods that I've talked about are contributed by them. We collaborated with, actually Brian Van Ness was involved early on, Carol for the breast cancer work, Nathan Pankratz for Parkinson's, which I haven't talked about those results. So a lot of people have been involved. And here's a broader list of that. Happy to take questions either now or offline.
So just for the breast cancer part, was it a specific, were they hormone receptor positive breast cancers? Is that what you're looking at?
It was a mix. It was a mix of different.
Because some of the things you picked up, like 19A1 is aromatase, which obviously is an estrogen pathway.
Yeah. No, it's a mix, and that's something that we haven't followed up much since Carol's interested in that, obviously. But I'd be happy to talk more if someone is interested in pursuing these results. I think that's one challenge that we have is we have a lot of exciting results across a variety of diseases. We have collaborations that have started, but really it takes a lot of focus to take to the next level. I'd be happy to talk more about that if you're interested in some of the specifics. Yeah.
Great presentation. This is a kind of naive question. You at the beginning you listed several diseases, including height or the diabetes, something like that. So there, actually, how many of them, what kind of disease is better explained in this way, or whatever we do in this way, some of them are really not explainable.
Yeah, I think we have too little data to say. I mean, I can say that. Let me go back. So I think we don't have enough data yet to really make a broad conclusion about that. We tried in germline risk of pancreatic cancer and didn't find anything. So that's the one disease that we have tried. Like there's some decent cohorts out there. We couldn't find anything. All these others, we can at least find a few pathways that are supported by these statistical interactions. I think it's too early to say, what determines whether we can find something or not. I think that there's interactions behind many of these. The extent that they explain all the rest of the missing heritability, I think that's pretty low. I think this is one of many factors that are an issue.
And when you draw the line in the meaning of SNP correlation, so how many of them you guess have a kind of mechanistic interaction? For example, some of the genes may affect, for example, let's say body weight, then may affect some genes like the one you showed there.
Yeah.
It means how we can better figure out the meaning of mechanistics behind this SNP correlation.
Yeah, so I think what we know from yeast is that many genetic interactions are going to be in combinations that do not have a physical association. That said, proteins that physically interact are highly enriched for genetic interactions. So a subset of these are, and probably the most interpretable subset are. I think that GATA3 pathway is interesting. So NFAT-C1, I don't know a lot about it. I do know that it physically interacts with GATA3. And so that's a case where the interpretation might be clearer because the reason these two SNPs interact might be tied to the fact that those two physically interact at the protein level. So I should say we expect many genetic interactions that don't have that mechanistic physical link, but the ones that do maybe are the places we should start. Good questions.
So you know our interest in probably why this question is coming, but as you identify these pathways, one of the things we also learned in the last 10 years is many of these malignancies are genetic variants that are agnostic in terms of tissue of origin.
Yeah.
Now, many of those mutations now have targeted therapies. So the question is, can you now take some of these pathways that are being associated and infer from that what some of the targeted therapies might be based on those pathways?
Yeah, I think that's the goal, and that's why we started this company because we thought other people would be interested in that too. So I think it's a long road. I can find statistical associations. It turns out that even getting the GWAS community to acknowledge that this is real, this is a paradigm shift, and you can see that in my grant applications, our papers.
But you did win the competition.
We did win the competition because we did something different, right? But I think so. That's obviously, if we find combinatorial genetic basis down the road, that should lead to new ways to target and very personalized ways to target. What this suggests is that people all have breast cancer potentially, or a subset have breast cancer because of the same phenotype, but they're very heterogeneous in the genetic mechanisms that got them there. But they should all respond to a therapy that modulates the level of that pathway that's affected. So in some ways, that the genetic heterogeneity, there's a light at the end of the tunnel because the pathway tells you what the phenotype is that you need to target. But yeah, that's exactly right. So I can tell you the set of individuals that have this subtype. Essentially, we can define subtypes based on the predisposition, the germline predisposition class. So I can give you a cluster that has this subtype that appears to be because of a combination. And in breast cancer, any one of these interactions might affect 10 or 20% of that population, the disease population. But when you add up all the ways that there are to cross those two pathways, it turns out to be a sizable number of individuals, not because they all share the same combination of variants, because they cross the same two pathways with presumably partially deleterious variants.
Maybe I missed it, but when you showed the association with the AML gene set,
Yeah.
were there other cancer types in pathways in your data too? And that's the one that came out? So like there's no association with colon cancer or liver cancer or other types of cancers?
It's a good question. So there are, like, for instance, KEG, so that particular gene set came out of KEG. KEG has a whole bunch of gene sets that are sort of disease gene sets. They have a Parkinson's gene set. I think they probably do have a colon cancer, and we tested that. That did not come to the top of the list. So it's not as systematically covering all the cancers that you might expect, but there definitely are gene sets associated with other cancers. This AML one is special in that cohort for sure. So and because of some of these interactions with rare A and a few other, RUNX1 is a major driver in that set. So that gene set's special for that particular cohort in breast cancer.
Because I was just wondering if you can do the opposite of what you did, which is that you take two cancer types that you think should have these pathways in common, but we don't really have some link to each other, but we don't understand why. Yeah. And then you can take all these mutations and all these SNPs and look for where it might be.
Yeah, no, that's really interesting. We have a number of collaborations that are going on like that, where you can take the same approach, define gene sets, but don't search everywhere. Search only for predefined gene sets. And they still might be big. And we still can do things like, we talked to Ruben a bit about this, take a founder single gene variant or set and ask what things modify this. So essentially we're changing the algorithm to not do all by all search, but to search for other pathways that modify a gene set of interest. And that's a good idea because it increases your power to discover. If you really care about that pathway, now I'm much more powered to see effects that involve only that pathway because I haven't tested everything. I've only tested this. It's the same advantage of focused single variant tests where people say only test these 100 variants. You have a lot more power to discover. So if you have a disease or a collection of diseases where there's cohorts available and you've got pathways in mind, this is a perfect. There's a sort of directed mode of this approach as well. Yeah, it's a good question.
Any other questions or comments? All right. Thank you.
Thank you.